it should activate the ERK pathway, since this ligand we expected was what activated CXCR4, and the ERK pathway followed downstream from that receptor. We found that 5 minutes after adding SDF, the ERK pathway was activated, was peak activated. But then we added AMD. AMD is a molecule that's supposed to stop CXCR4. It's supposed to block it and stop it from working. And then we added SDF again. So in this case, we expected the ERK pathway not to be activated, because the ERK pathway is again downstream from CXCR4, and if CXCR4 is blocked, the phospho-ERK pathway should not be activated. However, we found that it was activated to a similar extent as when we did not use the AMP. So this baffled us, but what did we conclude from this? So for a long time, the researchers thought that the CXCR4 receptor and the SDF1 ligand were monogamous. In other words, the SDF1 only bonded to the CXCR4 receptor and vice versa. However, recent research shows that another similar chemokine receptor called CXCR7 also binds to SDF1. Our results seem to indicate that CXCR7, not CXCR4, is the primary receptor responsible for metastasis. If so, this would explain our results. CXCR4 should not be expressed more in metastatic cells, instead CXCR7 should be. Also, since AMD is a molecule that only blocks CXCR4, we expect that the ERK pathway should also continue to be expressed once we add an SDF. And on the bottom, we see that the beta acting with our control to show that we loaded approximately equally. After doing Western blotting, we also used a technique known as immunofluorescence. And this was to actually visualize the physical positions of the protein inside the cell. So first, we took our cells, cell lines that we were interested in, and fixated them using a formaldehyde solution. Then we added primary antibodies, again to signal the antigens that we were interested in, and added secondary antibodies that were linked to a species-specific dye. Then using a special IF microscopy, we've shown ultraviolet light on the secondary antibodies, which emitted a glow depending on what protein we were interested in. Here, unfortunately, our immunofluorescence results were very inconclusive, and we were not able to see CXCR4 activation like we had hoped. But here are some results to show the concept behind immunofluorescence, what we may have expected if our procedure had gone correctly. So initially, at 0 minutes, this is before we add SDF. We found that, before we add EGF, sorry. So these are EGFR receptors. So the bright red signals the EGFR receptors, and as you can see, they're on the membrane of the cells. After adding EGF, 30 minutes after adding them, we find that the EGFR receptors, the bright red spots, have moved inside the cell. In other words, they've internalized, which means they've bonded to the EGFR receptor. 60 minutes after adding EGF, we found that they've internalized even more. But 2 hours after adding EGF, many of the receptors have either returned to the cell membrane or have been degraded inside the cell. So what did we conclude? Our results seem to indicate that CXCR4 is not responsible for the epithelial-mesenchymal transition. Instead, a similar protein, CXCR4, could be responsible.
Sorry, CXCR7?
CXCR7, yeah, could be responsible for metastatic potential. This brings us to further research. So we want to make sure that CXCR4 is in fact not responsible. So we plan to use, previously we used AMD, a molecule that blocks CXCR4. However, this protein can have it can interfere with other receptors. So we want to use a technique called small interfering RNA to completely block CXCR4. If we find that CXCR4 is in fact not responsible for EMT, then we want to focus on CXCR7 and discover its role in the epithelial-mesenchymal transition. We also want to try more transcription factors. In our experiment, we looked at snails, twist, slug, zeb, and SID. But there are others, such as E12, pictured here, that may play a role in EMT. In the more immediate future, we want to repeat our Western blots that we've done that show potential, such as those of ecadherin and phospho-ERK, to strengthen our conclusion. Okay, that's it. Yeah.